Thanks, Amy. We have a lot of force from the FDA here today. We even have some members of the FDA who are not speaking, but maybe we'll comment later once who's approving the drugs today. So our next speaker is Steve Lemery, also from the FDA, and he's going to now follow up and talk about regulatory requirements for site-agnostic indications, something we spoke about earlier today as well.
Thank you. As was mentioned, my name is Steve Lemery. I'm Associate Director of Division Oncology Products 2. I was on the review team for the pembrolizumab/Keytruda approval for the deficient mismatch repair application. How do I, green? Okay. So I thought I would anchor my presentation on the pembrolizumab approval and talk about some of the regulatory aspects, following the approval of pembrolizumab for the application. So just to background, in ASCO 2015, Young Lee presented the initial data on patients with deficient mismatch repair who received pembrolizumab. This was a small study initiated at Johns Hopkins, and I'll mention the landscape of GI approvals at that time. So I was the GI cancer team leader, and we had approved since 2011, which I started, around 3 drugs in late-line GI cancers that had modest effects on overall survival, about 6 or 7 weeks each. And the response rates was probably about 1% to 5%. Very few patients actually responded to these therapies as far as tumor shrinkage. They lived a little bit longer, but the tumors did not shrink. In that setting, we had about a 50-60% response rate in these refractory GI tumors, which was just not seen in GI cancers at the time. I mean, I was at the ASCO presentation that Young Lee did, and it was a profound sort of presentation to see these responses, including complete responses. Since that ASCO presentation, the approval of pembrolizumab, I've talked to a few patients, including one patient. She's a woman she had diffusely metastatic esophageal cancer, had a complete response for over 2 years on checkpoint inhibition therapy. She's not something you've seen, prior to checkpoint inhibition, in GI cancer. So it really was a profound effect that we'd seen at that time. So just given that we have a broad audience here, I quickly mention in a very simplistic way that mismatch repair deficiency is generally caused by mutation in one of 4 DNA repair proteins. It can be inherited as seen in Lynch syndrome, which is a cancer predisposition syndrome. But most patients with mismatch repair don't have Lynch syndrome. Sometimes they have inactivation of one of the DNA repair proteins. And this is usually diagnosed with IHC, or immunohistochemistry. So microsatellite instability is, in essence, the measurable phenotype of mismatch repair deficiency. And everyone has short repeats of these DNA segments or microsatellites. The links vary from person to person. And this MSI testing can report instability of these microsatellites. Usually it's detected with PCR or next-gen sequencing. So importantly, microsatellite instability is associated with increased tumor mutation burden, and it can be seen in many different tumor types. So this is off Young Lee's science paper. However, the red boxes I added in an orphan diseases presentation, where these really shows that there are some rare tumor types that have microsatellite instability, and really it would be impractical, if not impossible, to develop drugs in, quote unquote, these rare biomarker positive patients in these rare tumor types. So here's a partial development timeline of the application. So again, I saw the initial ASCO presentation in 2015. There was an update in 2016 where they had enrolled 53 patients at the time. And as you can see, about a year later, we approved the supplemental BLA. So it was a very quick development, both enrollment and development of this application. So as background, here's the data supporting the application, which was shown in a different format earlier. What was really important here is not just the response rate, but the durability of these responses. So as you can see, this was going out about 18 months at the end of the Kaplan-Meier curve. I think clearly that was based on a data cutoff, and some of these responses are quite durable. So again, I talked to a patient who had responded, complete response for over two years, really profound. The other important thing is that you have some of the other bucket was of different patients with different tumor types was mentioned in the prior presentation. A lot of the patients that had non-responses or non-evaluable actually turned out to be responses later. This was based on the data cutoff that we used for labeling. So I think my vague recollection was 13 of the 14 different tumor types had responses. So really you had responses in all these different tumor types. You might have slight variations in the response rate in different tumor types, possibly related to either chance or related to prior chemotherapies that patients received. Often patients with colon cancer had received more chemotherapy than some of the other tumor types. And so when we approved the application, we thought there was very strong biology. So as I think we've seen this figure earlier, one of the highest tumor mutation burdens is in patients with microsatellite instability. As you see, also melanoma is high up there, as is lung cancer. And so I think what we've seen in many of these different applications for checkpoint inhibition, as most of you know, is that response rates tend to be higher in patients with high mutation burden. And it's of the highest in patients with mismatch repair deficiency. So we thought there was a strong biology, there's strong clinical data. It was approved for patients without available therapy. So there's unmet need for these patients. We thought rather than requiring separate development program in each different tumor type, which would take years and years, if not decades, requiring patients to in essence, go into hospice, we approved it for all the patients with the strong clinical data. And there are post-approval requirements. Merck has enrolled over 400 patients with MSI high or mismatch repair deficiency to further assess the response rate in the different tumor types in patients with these tumors. So I'll briefly mention some of the unique developmental considerations, including pediatrics, in vitro diagnostic devices, disease versus indication, and some various other questions that we have. So, when we did the approval, we did approve it for pediatric patients as well, which I think is important. These are different examples of biomarker-positive tumors in different pediatric cancers. In MSI high, you have kids with congenital mismatch repair deficiency who have mutation burdens that are even higher than the adult patients. And at least anecdotally, we've seen profound responses in some of these children. Enteric fusion was mentioned earlier with the kids with infantile fibrosarcoma, and with ALK and ROS, you have various different tumor types as well. So as I mentioned, again, with immunotherapy, with gliomas, it generally has been a non-responsive tumor, but this is a case, actually this is not pembrolizumab, this was a case report of a patient, twins who had received nivolumab, who had congenital mismatch repair deficiency, and although rare, patients can benefit. And at a Stand Up to Cancer meeting, there was actually a separate image of a complete response to both nivo and ipi in one patient. The risk in these patients, of course, is CNS tumor swelling, and there has been another case report of a patient who died after receiving checkpoint inhibition. So I think you have to find out how to treat these patients best. So with in vitro diagnostic considerations, it was mentioned earlier, we did approve it based on the unmet need without a companion in vitro diagnostic device. However, there are post-marketing commitments to develop both an IHC and a PCR-based test. Optimally, I won't get into it in great detail, probably more than one different test will be useful for these patients. Again, when you're talking about very rare patient populations, that a test that can test multiple different biomarkers at the same time, like NGS, may be useful, especially if the prevalence of mismatch repair deficiency is rare in these populations. In other populations, like endometrial cancer, an IHC test that can be quickly done may be more beneficial. So this is just a paper that actually showed that testing does matter in these patients. So, in one of the tests that's out there that's used today in patients, there are differences in sensitivity for patients with colon cancer versus endometrial cancer. So it is important to know these differences to know where the test may fail, so you can perhaps test with a different modality if necessary in specific tumor types. So quickly, this is more of a philosophical question in our New England perspective. Actually, I think our initial proposal was is this a disease? But I think we later amended it to say this is an indication rather than a disease. So there are some arguments that MSI is one disease, including these tumors share histological characteristics, including lymphocytic infiltration and medullary type patterns, increased tumor mutation burden, and of course, a response to checkpoint inhibition. There are arguments against, however. This is probably not the only molecular finding in these patients. There may be differences in natural history or differences in responses to cytotoxic therapies, such that Folfox may work for colon cancer whose MSI high, but may not work for a patient with GBM. And it's hard to argue that baby with infantile fibrosarcoma has the same disease as NTRK-positive lung cancer. So I'll finally get into some of these development and regulatory considerations regarding tissue-agnostic development. So of course you're going to have, as was mentioned, uncertainty regarding effects on different tumor types. It was mentioned earlier, post-marketing data probably will be important to further assess this uncertainty. Again, even in randomized trials with different subgroups of patients, there's not certainty for a treatment effect in all patients, even in a randomized trial. I'll briefly mention, developing a tissue-agnostic setting could impact your trials of the drug in non-biomarker selected patients, and I'll mention that later. And I do want to mention that FDA has used the principles of tissue-agnostic development to support tissue-agnostic approval. So it's not an all or nothing phenomenon where if you don't get tissue-agnostic, you're kind of dead in the water. So again, Amy mentioned this in her prior talk. For dabrafenib, trametinib, for example, we've approved it in very small sample sizes in 2 indications where there is a high unmet need. So in the anaplastic thyroid cancer indication that I worked on, we had already approved these drugs for patients with melanoma, very high response rate, 60-70%, improvements in randomized trials. And so this was another setting where there was a high response rate, 60-70% in a very small patient population, very high unmet need, and we approved it based on those criteria. Again, this probably would not have been appropriate for tissue agnostic because of the colon cancer, because of the differences in heterogeneity in the different tumor types. However, we use some of these principles to approve it in these other settings. So, as this has been mentioned a little bit earlier, if you have a rare biomarker positive tumor, very, very high treatment effect on response rate, a randomized trial may not be feasible or ethical, or it may not be possible to do it in the refractory setting. And we did think that was the case with the MSI application. So although we approved it on single arm data and it was an accelerated approval, the full approval is to further assess response rate across the many different tumor types in their current trial, not doing a randomized trial. Merck is doing a randomized trial on colon cancer. However, that wasn't part of the PMR for the accelerated approval. So future challenges with tissue agnostic development, what if the biomarker is quantitative? Dr. Rizvi mentioned a little bit this earlier. So, you almost have 2 questions. How many mutations per megabase, for example, if you're going to define what the disease is, if we're calling this a disease, is it 10, is it 20, is it 30? What's the correct number? And how will different IVDs classify patients differently? So, if you have one company developing a drug for a TMB of 10, another for 20, and then each device, each next-gen panel classifies it slightly differently, you're going to have a big mess out there. And so this is a situation we can foresee in the future that is a challenge. And there is an effort that we are working with the Friends of Cancer Research to hopefully address this and standardize testing. Other challenges, combination trials. We've talked a lot about combination trials and assessing a treatment effect. It's hard enough with one drug, you add another drug on it, across all these different tumor types, it's a challenge. So, ultimately it's the company's responsibility to bring in a data package that shows that, both drugs add something to the treatment regimen to support approval. It was mentioned in the prior session also about, how to manage the label as new data are accrued, and this can be pre-market data, post-market trials, registries, real-world data. All these may influence the indication as more patients are treated. I always get when I've done this question at a different meeting, I always get how many tumors are necessary to support an application, and there's no right answer. I mean, clearly, if you have an application that you've only come in with 3 tumor types, it's probably not going to support tissue agnostic indication. I mean, if you have a lot of patients that have received the drug with those 3 tumor types and the response rate is 70% in those, well, maybe you'll support in those 3 tumor types, but it's probably not sufficient for a tissue agnostic indication. I think you have to make the case that the drug will work across the different tumor types, but there's not one right answer. So other questions we get are about the different pathways. What's enough for a tissue agnostic fast track designation, a breakthrough designation, accelerated approval? Fast track's challenging in this setting because it's often not just the effects of the drug, it's also the development plan. We approve the fast track development plan. So, when do we know the development plan is going to be sufficient to support tissue agnostic application. We're having current discussions internally about this. Same thing for breakthrough is more, you have more clinical data available to support a breakthrough designation. You need to have enough data to support that it's a breakthrough as a tissue agnostic as opposed to just in 1 or 2 tumor types. So key to all those though is unmet need, and that's what we had in the pembrolizumab application. So finally, how will tissue agnostic approval impact development for biomarker negative populations? So this is a theoretical example where you have a biomarker neoantigen positive tumor type that we approved the drug in. Should the patients who are biomarker positive be excluded from randomized clinical trials of single agent? I put checkpoint inhibitors. It could be any different PD-1, PD-L1, LAG-3, what have you. If not, you want to make sure that the improvement of an effect in the randomized trial is not driven solely by the biomarker positive population if we've already approved it in that biomarker tissue agnostic setting. So at a minimum, the biomarker should be identified in these trials and tested for. It may not matter if it's through a different pathway, like a cytotoxic drug, where the biomarker is not as integral to that drug. And as a hypothetical example, if you have a response rate of 50% in a tissue agnostic biomarker positive population, how would you conduct your randomized trial in a single cancer type, non-biomarker selected? It may depend on what the response rate is in the biomarker negative group, as well as what the prevalence or incidence rate is in that cancer type. If it's a very, very low incidence in that cancer type, it probably makes less of an overall effect in the entire randomized trial of all the patients. So in summary, we thought that the tissue agnostic approval of pembrolizumab facilitated faster access for patients with unmet need. We granted for every tumor type without all the tumor types being studied, but we did have at least 14 tumor types in the application. We did approve it for children with mismatch repair deficient cancers, and as part of the PMR, they're going to study the drug effects in children as well. We granted it without a companion in vitro diagnostic device, not advice. However, there are post-marketing commitments to develop these devices. And again, as you all know, it created new opportunities and challenges for drug development. That's it.